Welcome. Let us continue our discussion on excretory system. We have already come closer to the conclusion. A little bit was left. Today we will complete it. Now, what we have seen that the afferent arteriole, suppose this afferent arteriole is coming and forming lobes within the Bowman's capsule, forming glomerular tuft. This afferent arteriole breaks up into various lobes. And these loops, there I have made two loops. These loops ultimately joins to form efferent arteriole. This one is wide and short. This is wide and short. This is narrow and long. This arrangement has been made to keep the pressure, high pressure system, maintaining high pressure system here. In an injection you see from the greater diameter, if you send some liquid to the smaller diameter, the speed will increase. Now this apparent arteriole, we have seen that apparent arteriole is found to contain a cuff-like structure, cells. These cells are called juxtaglomerular cells. These are all juxtaglomerular cells. Now what actually happens? Suppose this one is the glomerular capillaries. These capillaries, I am making it a little bit a bigger one so as to enable us to understand properly. Suppose this one, this is a bigger one, this is a bigger one. So these bigger capillaries, this is endothelium, this is known as endothelium. These endothelial cells have pores inside, like this. Suppose this is endothelial flat surface, these are having pores. These pores are known as fenestrations. Or pores. These endothelial cells have fenestrations, that is, they have pores. These pores have a diameter of 50 to 70 nanometer. 50 to 70 nanometer diameter is there. Whenever blood is coming through here, so through these endothelial cells which have got fenestration, or these are all fenestrated as we say, it goes out through these pores. But the limitation is 50 to 60 nanometer. If it is less than 50 to 70 nanometer, then only it will pass out. And now there I will write the glomerular capillaries. This is glomerular capillaries, and these are all podocytes. Podocytes are like these are all cells. These endothelial cells have basement membrane. After this, there is basement membrane. Suppose this is basement membrane. So whatever we pass, it has to go through the basement membrane. Now the basement membrane is the only continuous layer, otherwise it has pores. And this also has pores, I will come. Now from these fenestrations, the molecules, the urinary waste, metabolic waste products, along with other water and some important items goes out. And it has a barrier inside. This barrier is known as basement membrane. This basement membrane under electron microscope has got a denser area in the middle. But whenever you see from this side and that side, it is lucent. It is lucent, that is, it has got some lucency, so it can pass. Now this is basement membrane through which the filtration or nephric filtrate comes out. Now whenever it comes out, these are all podocytes. These we call it as podocytes. These podocytes are actually, it is the parietal layer. This is the parietal layer of Bowman's capsule. This is proximal convoluted tubule. Proximal convoluted tubule. This is the parietal layer. Parietal layer. And these parietal layer cells is modified to form podocytes. These podocytes are food processes. These are the food processes. These are the food processes. And these food processes have pedicels at the end. Pedicels remain attached with the basement membrane. And they remain as such, that is, they are joining one finger with the other finger like this, but there remain some pores. This is a pore, this is a pore. These pores are known as slit. Slit or pores. These pores have a diameter of, if it is 50 to 20, this is 20 to 30 nanometer. So if glomerular filtrate has to pass through, because of the high pressure system here, 70 mm of Hg, it passes through the fenestrations of the endothelial cells or the glomerular capillaries. They pass on to the basement membrane. These are all some layers we don't have to go through. Lumina, rara, lumina, externa, this and that, these are names. Now it passes on from the basement membrane and it passes through the slit or pores. Slit or pores, and then the filtrate goes to Bowman's space. It is known as Bowman's space. Till then, after so many this barrier, that is fenestration is limited to 50 to 70 nanometer. Then there is a regular continuous layer of basement membrane, and from this basement membrane there is slit pores. From this slit pores, actually what happens, from this slit pores, it goes from here to here. So the pressure is minimized and it cannot pass through other than these 20 to 30 nanometer pores. These slit pores, from this slit pore, the glomerular filtrate comes and it comes as a glomerular filtrate, not urine, because it has some important materials which the body needs. So it has to be reabsorbed by this efferent arteriole making capillaries over this. They form this efferent, this is efferent arteriole, this is efferent arteriole. This efferent arteriole forms capillary network over the PCT, loop of Henle and distal convoluted tubule. This capillary is known as peritubular capillaries. So what we write in the exam? That efferent arteriole, after breaking down into glomerular tuft, and it anastomize and joins as efferent arteriole. Efferent arteriole has got some other functions to perform. That is, efferent arteriole, after coming out from the Bowman's capsule, it again makes capillaries. It may again make capillaries over the PCT, loop of Henle and distal convoluted tubule. From these tubules are known as peritubular capillaries, and these peritubular capillaries again joins to form vein. This is known as renal vein. That renal vein comes out. From here to a renal vein and renal artery taking blood, and this is ureter. So whatever is left behind after reabsorption, that is known as urine. In this way, the filtration takes place. Now there are 3 or 4 items or short points, short notes, which we want to highlight, but it will be worth mentioning that how these 3 accessory things helps. Or associated with the excretory system. Suppose if it is afferent arteriole, it is forming loops. And joins to form efferent arteriole. This is afferent arteriole. What we have come across, that afferent arteriole, when it remains in the glomerular, let us make a little smaller one. This is podocytes. These are all podocytes, inner one, or the visceral layer of the Barman's capsule is called podocytes. These are all podocytes, and this is the parietal layer formed of squamous epithelial cells. So these podocytes, and this, there I have shown that this forms basal lamina, and like that it forms, and the glomerular filtrate comes out here. Clear? This afferent arteriole found to contain what? Some important cough collar called juxta glomerular cells. And now this is the proximal convoluted tubule having brush border cells made for absorption. And then it forms, it is going towards the medulla, so it is known as descending limb of loop of Henle. This is the descending limb. This is loop of Henle. It is the ascending limbs. It is thin first and then it becomes thick. It becomes thick. So this is the thin, descending, ascending. Thick, thin ascending limb. This is thick ascending limb of loop of Henle. This is the thin ascending limb of loop of Henle. Now this loop of Henle again, when comes, what am I saying? Proximal convoluted tubule. They, this is proximal convoluted tubule. They again comes in contact. This is known as proximal convoluted tubule. This will be known as distal convoluted tubule. The distal convoluted tube, along its way, it again meets both the efferent and afferent arterioles. Efferent and afferent arterioles. It mixes. Let us make it a little bit colored. It mixes and it goes out. So here it is seen that they found to contain some cells with dense nuclei. These cells with dense nuclei is known as macula densa. Clear? So this is juxtaglomerular cells, and the distal convoluted tubule, while going to collecting tubule, it passes on through the efferent arteriole and afferent arteriole, that is in the glomerulus. Near the glomerulus is found to contain some dense cell with packed nuclei. And this is known as macula densa. Clear? Now, one thing more. In the joining of this afferent and efferent arteriole, here there is some cells which make it more rigid. These cells are called Polkition cells. Polkitions or polar cushion. Polkition cells or lattice cells. So what we will see, what we will write, that the aperture or the opening through which the afferent arteriole enters the Bowman's capsule and efferent arteriole both in the angle created by them, there remain some special cells called polar cushion cells or lattice cells. So polar cushion cells gone through, juxtaglomerular cells gone through, and again there are some cells in between this glomerular tuft. They are very important cells. These cells are known as mesangial cells. These cells are known as mesangial cells. These mesangial cells, and they have some connective tissue. They have connective tissue. Together they are called mesangium. Mesangium. They have got contractile properties. Contractile properties, that is, they can contract, they can expand, and they maintain the blood pressure here. Number one. Number two, they are phagocytic in nature. If anything comes in, they phagocyte those particles also. So these mesangial cells are form a network with the connective tissue called the mesangium. Mesangium. So three types of cells we have come across, that is juxtaglomerular cells, then Polkition cells or lattice cells, and here there is macula densa. Now, what are the juxtaglomerular apparatus? Known as juxtaglomerular apparatus is juxtaglomerular cells, these three, juxtaglomerular cells, lattice cells, and macula densa. This forms juxtaglomerular apparatus. They form juxtaglomerular apparatus. In this way, the kidney functions, that is the nephron functions in this way. Now, we will discuss a little of the problems faced by glomerulus. In the glomerulus, we will see glomerulonephritis is the condition where glomerulus is having inflammation. Inflammation of the glomeruli. It's called glomerulonephritis. Again, glomerulosclerosis. This is a condition where the glomerular tuft becomes rigid. Glomerular afferent arteriole, glomerular tuft or glomerular capillaries becomes hard. So if the glomerular capillaries here becomes hard, it cannot pass out the urine, it cannot filtrate. It is a very dangerous disease. It may cause kidney failure or renal failure if not treated earlier. It is called glomerulosclerosis. Sometimes diabetic related neuropathy. That is because of the diabetes, some nerve gets infected or nerves gets destroyed. So in that case also renal failure may occur. Renal failure can cause by diabetic related neuropathy. Glomerulosclerosis is glomerular capillaries become hard, and glomerulonephritis, inflammation of the glomeruli. These are the 3 or 4 diseases, and sometimes we have heard of kidney stones. Sometimes we have heard kidney stones. Kidney stones are actually oxalate crystals. Oxalate crystals are found in the urine. If the urine found calcium oxalate, say for example. So many fruits, vegetables and intake of maximum calcium oxalate may cause these kidney stones. It is found in urine, and the doctor prescribes the medicines to dilute this calcium oxalate. Now at the last point, we'll discuss about uremia. Uremia is a condition in which the blood got maximum urea inside the blood. That is, proper filtration does not take place. That is called uremia. This uremia causes kidney failure, and the treatment is hemodialysis. Hemodialysis means dialysis. How it is done? It is from artery. Usually now it is done from the brachial artery. Brachial artery is in the heart. You will see whenever it is going out, it gives out subclavian, carotid, internal carotid, so many arteries. So nearing the artery here, that artery is pierced. Artery is pierced, and the blood full of urea, that is, patient is suffering from uremia, this artery is pierced and the blood is taken into a apparatus called kidney, artificial kidney. The blood is taken in here and it is mixed with some anticoagulant. Some anticoagulant so that the blood does not coagulate. Anticoagulant is mixed and then it passes through this apparatus. This apparatus have cellophane. This apparatus have membrane, permeable membrane called cellophane. From this cellophane, the urea and uric acid cannot goes in, and the good blood, which is devoid of urea, comes out. Devoid of urea, it is coming out. It comes out from this machine. This good blood, that is less urea or not urea at all, it comes out from this apparatus or artificial kidney. From this artificial kidney, now this blood is made what should I say, made to match with the body temperature. So it is in 0 degree centigrade, here it is reduced to 0 degree centigrade, and if this filtration takes place outside the body, and whenever it goes out, it made to match the body temperature. It is again anticoagulant, and then some coagulant is added. Antiheparin, coagulant is added so that it can maintain the coagulity, normal coagulity of the blood. Because we are not patient, we are having some anticoagulant factors inside our blood which makes the blood does not clot. So some coagulant is added. Here anticoagulant was added. Now a amount of coagulant as per the conditions of the body, some coagulant is added, and this artery, this will go to, they are draining it some vein. They are draining it into a vein so that good blood, that is less urea, is maintained. So this is known as dialysis. In this way the dialysis takes place. This kidney is very, very important. Please go through the entire details of it so that you will be able to learn the total about this excretory system. Till then, thank you very much.